Good afternoon, everyone, and welcome to the Office of the Chief Scientist Science Seminar Series. I'm Andrew Paul, and I'm a science advisor with the Office of the Chief Scientist and have been your host for this fall's series of seminars. I'd like to start by respectfully acknowledging that in Alberta, we are on the traditional gathering places and home to people of Treaty 4, Treaty 6, Treaty 7, Treaty 8, Treaty 10, and the Métis. Wherever you find yourself today, I encourage you to recognize and acknowledge the relationship that First Nations, Inuit, and Métis have with the land upon which we all live, work, and learn upon. These science seminars provide an opportunity for us to get together to exchange knowledge on interesting science that's happening throughout Alberta and do it in an open and learning environment. The hope is that through our discussions and presentation today, we are able to improve our awareness and understanding and to put that knowledge into practice through evidence and informed decision making. In order to keep the quality of video and audio working today, your video that's been disabled, but to ask questions or make comments, please enter those into the chat box at any time during the presentation. I'll hold those questions or comments until after today's presentation, and then we'll read them out in the order that they were received. Hopefully we have time to get through all of them. If not, if we can't get through all of them, we'll certainly provide those questions and comments to today's presenters after their talk today. And with that, I would like to introduce our speakers today, and we are welcoming Jessica Reilly and Dr. Joshua Miller, and they'll be presenting on genetic and morphological diversity of Arctic grayling in Alberta. Joshua is a molecular ecologist interested in using a wide range of genetic and genomic techniques to inform management and conservation of wild species. He applies methods in molecular ecology and genetics to complement more traditional ecology that together form the basis of a more holistic management strategy that can conserve long-term health of populations. He has applied this framework to bighorn sheep, Galapagos tortoises, sorry, I almost said that, polar bears, and Arctic grayling. Dr. Miller is an assistant professor at MacEwan University in Edmonton. And as well, we have Jessica. Jessica's an avid angler that grew up in Alberta fishing the East Slopes with her dad. Throughout her career, she has worked at the interface of science and management, integrating research findings into practical applications for species status assessments, native trout recovery, and monitoring programs for river fish. Jessica specializes in applied conservation genetics and cumulative effects management within Alberta environment and protected areas, where she has worked as a regional fisheries biologist. She's worked with the Species at Risk team and currently is the fisheries manager for Southern Alberta. Jessica is also a member of the Freshwater Fishes Scientific Subcommittee for COSEWIC, which is the Committee on Status of Endangered Wildlife in Canada. And with that, Jess, I will turn it over to you to start presenting.
Great, thanks, Andy. And thanks to everybody joining Dr. Miller and I today to talk about Arctic grayling and their morphological and genetic diversity. So Arctic grayling is part of the genus Thymallus, which is a group of fish best known for their large and flashy dorsal fins. And there's been a fast-growing body of literature describing substantial diversity within this group over the past 20 years, with many new species and subspecies designations being proposed. But depending on who you talk to, there could be anywhere from 13 to 15 different species, and so the taxonomy of this group is far from resolved. And even just thinking about the grayling that we have here in Alberta, there could be higher levels of intraspecies variation than historically assumed. So what I'm showing you here is a time-calibrated phylogeny of T. arcticus based on the whole mitogenome. And sorry, I didn't mean T. arcticus, I meant Thymallus. But what I'm showing you is a snapshot of T. arcticus on the top and then a closely related species, the Baikal grayling, on the bottom. And these two species diverged from each other around 5 million years ago, so in the Pliocene. But what's interesting is there is another divergent node within T. arcticus from around 3 million years ago. And just for a little bit of context, that's about the same level of divergence that we see between two different species of cutthroat trout, West Slopes and Yellowstones. And that represents many, many, many generations of Arctic grayling traveling down their own evolutionary path. In North America, we've called those two groups the Beringia and Nahanni lineages as a nod to where we think they survived the last glacial period, Beringia being in current-day Alaska and Nahanni just north of Alberta. And when the ice sheets retreated, fish from these refugia colonized outwards, and researchers from UBC were curious, where did their descendants end up? And they noted that the Beringia fish were found throughout the like Alaska, Yukon, BC, down into Montana and Saskatchewan. So that's shown with the black and hash symbology here. And that the Nahanni fish were found to be quite localized to the Mackenzie River drainage and the Liard River watershed. But quite a bit of the species range wasn't included in this study, including Alberta. So fast forward 10 years, and I was doing some research on Arctic grayling, looking at a different molecular marker called microsatellites. And I observed this really interesting spatial pattern in mutations at two loci. And I always saw these mutations in Hay River fish. I saw them about 50% of the time in Peace River fish, and I never saw them in fish from the Athabasca drainage, even though we sampled almost 1,000 fish. And although it's not odd to see mutations in occasional fish, this kind of striking spatial pattern is a clue that there is some deep historical divergence driving it. And so it made us ask the question, hey, maybe we have these two phylogeographic lineages here in the province. Unfortunately, Dr. Miller was also interested in resolving this question, and so together we put in the next puzzle piece on the Grayling phylogeographic map. And we found that, yeah, indeed, we do have both lineages in the province. The Beringia fish are found throughout the Athabasca and in the Peace, whereas the Nahanni fish are found up in the Hay and also in the Peace. And you'll see that there are some streams that have both lineages. And so because we see such substantial divergence in the mitochondrial DNA variation, it just really begs the question, do these fish differ in other ways? Do they look different, behave differently, and do they interbreed? And so Josh and I started tackling that next mountain of questions by asking, are there morphological differences between Nahanni and Beringia fish in Alberta? And so we looked at body shape, meristic traits, which are body parts that you can count, like fins and scales. And then we use mitochondrial DNA to assign fish back to their lineage. So what might we see in terms of grayling morphological differences? So salmonids are a phenotypically diverse bunch, and there is many examples from other species of significant differences within species in terms of body shapes, how streamlined they are, shape of their fins, shape of their heads and jaws. And the image I'm showing you here is kind of a classic example from lake trout, where these are all called lake trout. They're actually all from the same lake, but their body shape is really different, and also their head and jaw shape, depending on what habitats that they're found in the lake. And these differences aren't just cosmetic. They do translate to real differences in performance in terms of how well fish can swim, depending on, if are they in a fast flowing system or are they in still waters, and also what kind of prey species that they can capture. So to find Arctic grayling for our morphology study, we traveled up to the Caribou Mountains, which is in the northern part of the province, and hoping to actually find the Nahanni fish. And then we did a big tour of northern Alberta and the Peace and Athabasca, hoping to find Beringia fish. And we did have success in the Little Smoky and Freeman River systems. All the fish were captured by volunteer anglers. And this is an incredible group of people that not only fished hard for us, but also carried hundreds of pounds of gear up and down riverbanks and gave us their secret fishing spots so we could catch enough fish for the study. The genetic analysis was done at MacEwan University. And then we used geometric morphometric techniques to analyze body shape. And this approach requires far more than a snapshot from your iPhone because photographic distortion can introduce a lot of error into your data sets. So you'll see in this photo here, we have specialized camera equipment and specialized cradles, etc., for the fish to be held in. And then we counted 12 meristic traits. So here we have a volunteer biologist, and she's counting something called phylogeographic, which are these finger-like projections in the intestines of the fish, and these are thought to increase absorptive capacity. And we were also very fortunate to find a veterinary clinic in Edmonton that allowed us to take x-rays of some of the Arctic grayling so that we could count vertebrae. So what did we see? Considering 16 landmarks, we did observe differences between the lineages in the snout relative to the eye and the width of the caudal region. And because there were these differences, we were able to correctly assign about 95% of fish to their genetic lineage based on body shape alone. So this can be interpreted as subtle but significant differences between the lineages. In terms of meristic trait results, we found differences at 3 out of 12 traits. So those are scales along the lateral line, branched pelvic rays, and pyloric caecae. It is important to note that none of these traits are diagnostic, so you'll see that there is overlap between counts for all of these traits. Another really cool claim to fame for Alberta grayling was that we observed the highest scale line counts for any grayling species ever recorded. So some of those Nahanni grayling are very, very scaly bunch. And if you are interested in learning more about this study, we have a recent publication that you can check out. So this kind of morphological work can seem like hair splitting, but it is really important to understand the overall intraspecies variation that we see in Arctic grayling. And interspecies variation is linked to like long-term species resiliency. Diverse species, there's a higher chance that some individuals will be able to survive through changing environments. And also if some of that variation are heritable traits, those aren't easy to recover if they're lost. And so for a species in decline like Arctic grayling, that becomes really important information, seeing that we've already lost some populations from the province. Now, the importance of interspecies variation is acknowledged by many jurisdictions, including Canada. And it's done through recognizing units below the species level. And in Canada, they're called designatable units. And these units are meant to be discrete and evolutionarily significant, meaning that these groups contain heritable adaptive traits. And in practice, what this enables us to do is divide the species range into smaller pieces. The pieces can be assessed independently, and assessments can produce different status ranks. So some designatable units can be at risk and others secure, which can lead to different levels of preventative measures or management interventions, depending on the needs of that unit. Now, it is challenging to do this step for a data-limited species like Arctic grayling. So it's often used as a weighted evidence approach where evidence is considered at the same time. So that could be things like genetic data, morphology data. And one of these approaches was published by Jonathan Mee a couple years ago. And it's kind of interesting to see where Alberta Arctic grayling line up when considering this approach. So one of the first kind of categories of information that they consider is taxonomic entity. Are there separate named species or are populations isolated? And for Alberta Arctic grayling, it's a no. They're all called Arctic grayling. And they aren't technically isolated. They're all in hydrologically connected systems, although sometimes movement between could be impacted by things like water temperature. Do we see diagnostic lineages? Absolutely. That's the Nahanni and Beringia lineage. Do they live in different ecoregions? So presence in different ecoregions is a way to infer local adaptation because the assumption is fish that live under different climatic regimes or with different kinds of species assemblages might have different traits. But for Alberta Arctic grayling, they actually all live in the same ecoregion, which COSEWIC is called the Western Arctic ecoregion, shown here as number 13. And then how about actually local adaptations? So when we don't have perfect understanding of the very complex interplay of phenotype and genotype, what you can do is look at suites of traits and then consider, well, have those traits been shown to be at least partly heritable in other species? And that's kind of where our morphology study fits in. We did see morphological differences between the lineages, but there are limitations to our study. So one of them is we weren't able to look at differences between the dorsal fin and coloration of Arctic grayling. And in other Thymallus species, that's actually one of the key diagnostic traits. We also only looked at a piece of the Arctic grayling range, and so we're not sure if the variation that we saw is truly representative of the variation within both of these lineages. We also don't know yet if the lineages hybridize with each other, and if they do, and we sampled some of those hybrids in this study, then that could have complicated our ability to distinguish those parental lineages. And then instead of looking at morphological traits and then not being totally sure if it's phenotypic plasticity or if it's heritable, why don't we just look at differences in coding genes between the lineages? And I'm really happy to say that we are moving forward on some of these questions. And with that, I'm going to pass it over to Dr. Miller, if I can figure out how to give you control.
Awesome. I seem to have control. So thank you for all of that. And I get the really fun part now of talking about what our next steps are. If it will go. Nope. Jess, you might have to advance for me.
I think I actually have to give you control.
Oh, okay.
Let's see. Nope, that didn't work, but I'm here. I will advance for you.
Sorry, everybody. So let's talk about next steps. So one of the first next steps we were tackling is to really get a wider assessment of genetics across the range. So Jess had up this map earlier, and it really highlights that sampling gap in the northern and eastern portion of the range. So one of the first things that we've done is really tried to contact as many people in the field as we could think of. I think we reached out at this point to over 30 individuals in government and local biologists, and that has been successful in some regions. Next slide. So thanks to the work of a really, really hardworking undergraduate, Morgan Warwa, here at MacEwan University, we have a much wider assessment of that mitochondrial genetic diversity across the range. So we have additional samples from the Yukon, including very far north in the Yukon. So these are, the new points are highlighted in red here, as well as from eastern Saskatchewan. Really interesting from this work, those northern Yukon samples have actually shown a new contact zone between Beringia and Nahanni lineage. So this was not known before and really expands the potential location for those Nahanni fish and perhaps colonization routes. We are still actively looking for samples as this work is ongoing. But one thing that has come out of it, and if Jess could go to the next slide, is that as part of her work, Morgan developed a new extraction protocol to get DNA out of a sample type that might have been completely overlooked if not for this work. And this is actually using scale samples. So researchers from Saskatchewan were very kind to send us a box of these scales that have been sitting at ambient temperature since the early 1980s. They were originally used as a way of aging fish from several locations around Saskatchewan, but hadn't really been touched since then. So Morgan was up for a challenge and tried to get DNA out of these scales. Next slide. So this picture here shows a comparison of different extraction protocols and different tissue types. So that panel A shows what extracted DNA looks like from fresh tissue. So this would be like the fish that we caught as part of the morphological analysis. It shows a very dark band way at the top of that picture, indicating that it's a very large pieces of DNA, and there's a lot of them. Panel B is showing an original extraction protocol applied to those scale samples. And what you can see is we don't see that really big dark band of DNA. There's kind of that lighter smear of really small fragmented pieces. And that's not really useful for analyses. It's very hard to work with those. But through troubleshooting that Morgan did and a new protocol that she helped to optimize, panel C shows DNA from those scale samples from the 80s. And you can see a much more robust amount of DNA. It's still fragmented. There's nothing we can do about that. It is from the 80s, but we had enough DNA that the sample results from Saskatchewan that were shown on that original graph all came from these archived scale samples. So it's a really exciting possibility that we could get additional samples without actually having to go out into the field using these archived and perhaps overlooked tissue types. So in addition to getting a wider sample from tissue, one thing you could do is apply what's called environmental DNA methods. So environmental DNA is a process where you're getting DNA from an environmental sample without having to interact with the organism that you're interested in. So here in this picture, we have a fish that's swimming around in water through natural processes. It's losing scales, it's losing mucus. And so that DNA contained in those samples is out in the water. So what we would do is put that water through a filter. So that's showing that next part of this figure. And then if you do a DNA extraction of that filter and all of the DNA contained in it, you might be able to detect species without actually having to physically see or interact with them. Next slide. So Melissa Matsoukas and Greg Goss, both at the University of Alberta, developed a assay to do this kind of eDNA work, environmental DNA work, for Arctic grayling. And they had what's known as an original assay. So this was a version 1, if you will, of an assay that looked for presence or absence of Arctic grayling. This is based on a technique called qPCR or quantitative PCR. And through a lot of troubleshooting, they show that it worked in field conditions. So you can see this researcher here is out taking a water sample from a water body, putting it through the filter. And then if you were to do that DNA extraction and apply the qPCR technique, it could detect grayling in areas that they were also known to occur through field assays. So this opens up a new method for detecting presence and absence of grayling in water bodies without having to do intensive field surveys. But this leaves a little bit to be desired in that we know that there's those two different lineages of Arctic grayling, the Beringia and Nahanni. And so Greg and Melissa worked on another update to that assay, which would be lineage specific. So this is taking advantage of the fact that we know there are these fixed differences in the DNA between Nahanni and Beringia fish. So that's shown here in this red box, that at certain positions in their mitochondrial DNA, Nahanni fish have a certain base pair and Beringia fish have a different one. So in this assay, they took advantage of this and developed probes. Am I going to the next slide? That would be specific to one lineage or another, or the other. And so in these graphs on the right side of this slide, you can see that top one is looking at the ability of the Beringia probe to detect a Beringia fish and not detect Nahanni fish. So what you see is that as we go forward in time, you can think of cycle as time, we're detecting just Beringia fish and we're never detecting Nahanni fish. And same thing for that Nahanni probe. That Nahanni probe is only detecting Nahanni fish or Nahanni DNA in the eDNA sample and never detecting Beringia DNA. So they analyzed these new probes in a bunch of positive controls, and it shows promise. A little more work needs to be done before it's deployed broadly, but it's a next step that we can go beyond presence absence to detecting these lineages using eDNA methods. So while all of this is really, really informative and very exciting, it's all so far been based on mitochondrial DNA, except for that little bit that Jess did as part of her master's work using microsatellites. And mitochondrial DNA is very informative. It's really been a backbone of a lot of population genetic and conservation genetic work, but it has a large limitation in that it's only maternally inherited. So by looking at only mitochondrial DNA, we're missing a lot of information that could be gleaned from both the matriline and the patriline. So one big thing that I'm working on, sorry, next slide, is to look at nuclear DNA. So nuclear DNA is DNA from the nucleus instead of from the mitochondria, and nuclear DNA is biparentally inherited. So you get information from both the matriline and the patriline. And so what I'm doing specifically is I've been working with, sorry, next slide, the Canada Biogenome Project to establish a reference genome for Arctic grayling. The Canada Biogenome Project is a consortium of research institutes that are trying to develop these high-quality reference genomes for a bunch of Canadian species. And they were very gracious in accepting that Arctic grayling should be one of them. So we have a draft genome already developed. Down here at the bottom of this graph is, bottom of this slide is a picture of what a genome would visually look like. This is not the Grayling genome, but it's just one for visual purposes. And what we're trying to get are these very long pieces of DNA all organized into these chromosomes. And so we have that with Arctic grayling. I'm not going to bore people with numbers that they might not care about, but I'm happy to go over that in more detail at the end of the talk if people are interested. But why is this useful? What are we going to do with it? So once we have that reference genome, we really can use it as a point of comparison. So what I'm hoping to do as part of my research program is what's called whole genome resequencing, or taking DNA from a bunch of different fish, in this case, sequencing their genomes, and then comparing it to the reference and comparing it to one another. So that's what's shown in this figure. Each one of those tubes would have DNA from different grayling individuals, and then we can compare those DNA sequences to the reference genome. And when we do that, next, we can start to investigate things like checking for hybridization, looking at population structure, and detecting those signatures of selection. So the problem with using mitochondrial DNA up until this point, as was highlighted by Jess earlier, is that we don't know if those two lineages are hybridizing. The nuclear genome would tell us that. And then that population structure piece is really, really important for determining the scale of conservation, seeing which water bodies are connected to one another, at what scale do fish migrate from one point to another. And then those signatures of selection, if we go to the next slide, is going to be really important for determining those DUs. So as Jess was mentioning earlier, We can use phenotypic traits to look for those signatures of selection, but we could also potentially use genetics as well. So this is a paper that I'm highlighting that has applied this kind of idea to salmon, Atlantic salmon, and trying to establish a sorry, my brain, how we can separate them into those designatable units and manage at an appropriate scale based on local adaptation. Next part of the slide. So this is kind of a conceptual diagram of how that works. You first look for those genetic differences on a genome level scale, and then see, are there parts of the genome that seem to be associated with those differences? You can then plot those differences on a map and see, in this case, all of those yellow points are at the north, all of the blue points are at the south, and then look for climactic or other variables that are associated with those breaks and see, is there a driver of that local adaptation, and use that to inform those designatable unit categorizations. So this is probably way into the future, but it's a place that I hope we are able to go with Arctic grayling. Next slide. So in summary, the work we've done and have ongoing has shown that there's a really large amount of intraspecific genetic and morphological variability within grayling, even just in Alberta. In North America, we have those two distinct lineages, and those morphological characteristics can diagnose them, even if it's subtle. And then we're really, really working to continue to build upon and expand consideration of Arctic grayling genetic diversity in hopes of making conservation and management decisions. And I just like to end with the fact that all of this work has been extremely collaborative. None of it would have happened without the generous contribution of funders, including the Alberta Conservation Association and NSERC, as well as in-kind contributions from the Northern Lights fly fishers and members of the Alberta government that have helped with so many things. So I appreciate all of them.
Excellent. Well, thank you both so much for a great presentation. We should start wrapping up now. Thank you everyone for joining today's science seminar. I want to remind you that our next seminar is not in 2 weeks. It's going to be next Tuesday, so 1 week from now on the 16th. And we have Dr. Holger Willie, who will be presenting on developing a vaccine for chronic wasting disease. So again, that is next Tuesday, December 16th. If you're not on the mailing list for these science seminars, please send an email to epa.ocs@gov.ab.ca. And with that, I'll end our presentation today and thank Josh and Jess for a wonderful presentation. Thank you both.
Thank you.